So I have received a few follow-up questions regarding the consequences that rhesus factor incompatibility may have, in particular leading to hemolytic disease of the newborn. This is a potential complication that happens when we have rhesus negative mother and a rhesus positive father. The rhesus factor that we are considering is the most antigenic, the factor D. So when we are talking about the rhesus factor here, we are really talking about the factor D rhesus factor. Now, when we think about the red blood cells, you remember the ABO blood grouping antigen system on the surface of the cells. And with the ABO blood grouping, there are naturally occurring antibodies in the plasma. These were our immunoglobulins. The rhesus factor, which is present on the surface of the red blood cell, we can represent that here as this kind of a block-like structure. This protein is either present or absent. So hence it's present in this particular cell. This means that it is rhesus positive cell in this case. But as you can see here, there are no naturally occurring antibodies to the rhesus factor in the plasma. But now, if the rhesus positive blood is mistakenly given to someone who is rhesus negative, let's say by a blood transfusion or from a fetus, we give that to someone who has rhesus negative blood, so they have no rhesus factor. Well, in this case, they normally have no rhesus factor antibodies in their plasma either. But now, if they are exposed to rhesus factor antigen, seen as these small gray blocks in this diagram on the surface of the rhesus positive red blood cell, well, they will become rhesus factor immunoglobulin type Gs. They will produce IgG antibodies. In reality, these are going to be Y-shaped molecules, like here, with a specific receptor for the rhesus factor present at the end of the immunoglobulins. So these rhesus factor antibodies, IgGs in this case, will only be produced by a recipient who is rhesus negative and who is exposed to rhesus positive blood. In a sense, that is the physiology that we're talking about here. Now, let's think of a situation where there is a rhesus positive father and a rhesus negative mother. This means that depending on the genotype of the father, there is either 50 or 100% chance of the fetus being rhesus positive. So whenever there is a rhesus negative mother and the father is rhesus positive, this is a potential complication that we end up considering that the baby might be developing a hemolytic disease of the newborn. Now, let's consider what is it that is going on in this case. So here we have a baby in the uterus of the mother, and the mother is rhesus negative, and she is carrying a rhesus positive fetus as a result of that dominant rhesus gene provided by the father. Now the risk is that we get fetal-maternal hemorrhage. This is blood leaking from the fetus into the maternal circulation. Now, this does not usually happen during the first part of the pregnancy, but it can happen towards the end of the pregnancy. But during the delivery, it is guaranteed that some of the fetal blood via the torn placenta is going to get into the mother. So in a first pregnancy with a rhesus negative mother, in this case the mother will not have any rhesus factor antibodies as long as she has not been exposed to any rhesus positive blood in any time in the past. But now, if she carries the rhesus positive fetus, and some of the rhesus positive fetus's red blood cells will get into the maternal circulation, some of the baby's red blood cells through this fetal maternal hemorrhage will get in. And of course, these babies' red blood cells are rhesus positive. And as a side note, as I said before, this will certainly happen during the delivery if it has not already happened during the later stages of the pregnancy. Now, what this will mean is that the mother will make these rhesus factor antibodies. In other words, the mother will become sensitized to the rhesus factor. So she will make rhesus factor antibodies, which previously she did not have. So now this baby is going to be born, and during this first pregnancy there is not usually any problem because most of the sensitization is likely to take place during the birth of the baby. So by the time a mother develops rhesus factor antibodies after the delivery, it's okay because the rhesus positive baby has been already delivered. What this means is that the mother is now sensitized to the rhesus factor. She now has rhesus factor antibodies because of the earlier fetal-maternal hemorrhage. Essentially, the baby's red blood cells are getting into the maternal circulation. Well, the baby is gone now. They are born. It is absolutely fine. But the mother can become pregnant for the second time. And again, it is likely with the rhesus positive father that there is another rhesus positive fetus that results. So this is the second child. Now, because of the mother having these rhesus factor antibodies in her circulation, so these are these Y-shaped IgG antibodies. And she's going to have millions of these in her circulation. I just drew a few. The problem that we face here is that the transplacental migration of antibodies does occur. And in fact, it is essential that it does occur because when the baby is born, we want the baby to have passive immunity as a result of the mother's antibodies. So this protects against all the bacterial and viral diseases that are around. But the downside of having rhesus factor antibodies is that the rhesus factor antibodies can migrate into the baby's circulation. And what will happen here is that these rhesus factor antibodies will start to agglutinate and hemolyze with the fetal red blood cells. Now, what does this mean? Well, the baby will develop hemolysis, and that is going to make the fetus anemic. So that is going to reduce the oxygen delivery to all parts of the baby's body. As well as that, as reduced oxygen carrying capacity of the blood, the fetal heart will try to compensate for that. And that can lead into the fetal cardiac failure. And even further, when the red blood cells are broken down, when the hemoglobin in the red blood cells is broken down, what is going to be produced? Well, a bilirubin is going to be produced as a hemoglobin breakdown product. But now we have all this hemolysis of the fetus. So we are going to get lots of bilirubin released. And bilirubin is particularly toxic for neurological tissue. So it's going to damage the brain development. So the brain of the fetus will not develop properly. As well as that, in the fetus, the red blood cells are produced by the liver and the spleen. So we are going to get enlargement of the liver and spleen as they try to produce more red blood cells. And as this develops, the liver gets bigger. The liver function is also going to be reduced. So the fetus is going to have reduced liver function. And the albumin is made in the liver. And this albumin is responsible for generating oncogenic pressure in the blood. And it is this oncogenic pressure that generates the osmolarity in the blood, which is going to suck the tissue fluids back into the intravascular component. So because the baby is going to be hypoalbumin anemic, because they are not now producing albumin, this has serious consequences. So what we are left with is a delivery of an anemic baby with high bilirubin, a risk of brain damage, and of course the child is going to be jaundiced because of the bilirubin as well. In more severe cases, there can be intrauterine death. Now, this is recognized in modern days. Some practitioners will perform intrauterine blood transfusion, which of course is a very technical procedure. But there are times when it is enough that the baby will receive blood transfusion after the birth. But it is rather hard to undo any damage caused by the bilirubin and the brain damage. So what we really need to do is to prevent this condition from happening. Now, if the first pregnancy, let's say we have said that some of the fetal factor antigens are going to get into the maternal circulation. Now, if there was a lot of antibodies there already, say we put them in there, let's say that we gave the mother an injection of anti-D immunoglobulin, then as soon as the fetal cells came in from the baby, they would be mopped up by these antibodies that we have injected. So that is exactly what we are going to do. A lot of practitioners will give an injection of anti-D at about 28 weeks into the pregnancy. Giving the anti-D antibodies is really a form of passive immunization of the mother. And we want to do that also immediately or as soon as possible after the birth too. Usually this takes place within the first 24 hours. If done, this would mean that any of the fetal red blood cells that are, in this case, of course, Rhesus positive, that have gotten into the mother, they will be quickly agglutinated and hemolyzed by these antibodies that we have given. But this is a passive form of immunization. Within a few months, all of these antibodies will have left the mother's body. And the key thing is that she will not have produced any of her own. So, by essentially giving the anti-D, we are giving these anti-Rhesus or anti-D immunoglobulins. And these are mopping up, a bit like a sponge, all of the fetal red blood cells, getting rid of them very quickly. I would say that this is probably within hours. All of these fetal red blood cells that would have otherwise been a concern have been collected and broken down. This means that the time when the mother is exposed to the rhesus factor antigen is actually very short, and she will not have time to produce her own antibodies. This means that for the second pregnancy, the mother will not have become sensitized and there is no risk for the fetus, as the mother will not have the rhesus factor antibodies. And therefore there will not be able to migrate into the fetus, and therefore the fetus will not develop hemolytic disease of the newborn. So this is really a classic example of where prevention is better than the cure. So what you should do is to suspect this possibility whenever we have a Rhesus positive father and a Rhesus negative mother, and give the anti-D according to your local protocols, probably at 28 weeks of the pregnancy, but certainly within the first 24 hours after the delivery. And that means that you are potentially saving the life of any subsequent fetuses, and I'm sure they will be very grateful for that.